Hello. Transcriptional initiation is complex in eukaryotes. Say if this is the promoter, and then formation of an initiation complex is easy relative to imagine if transcriptional or complex is formed and it has to move through the on the chromosome. This is especially difficult because if the DNA is wound around nucleosomes, that means it should keep on dislodging the nucleosomes or histones from while it moves and then make them behind. And in that there are several other ways how transcriptional elongation is complex. Let's have a look at some of these things as much as we could understand. First thing, of course, is to somehow recruit the elements that are involved or responsible for the transcription elongation itself. So we have the recruitment of a kinase PTFB, which also is responsible for the phosphorylation of the serine in this motif YSP-TSPS motif that is present on the carboxyl terminal domain of RNA polymerase. And that is a signal. If the second serine is phosphorylated, then there are several other factors that are elongation factors, which are similar to that of NUSG in bacteria, are recruited to the RNA polymerase, and that would allow the elongation to happen faster. One thing is to make the RNA polymerase move fast. Second thing is to dislodge the histones that are upstream of towards in the direction of the RNA polymerase. So there are these HSP5 and TAD-SF1 are elongation factors that act on the RNA polymerase to move faster. And there is yet another RNA elongation factor, that is TF2S, which apparently reduces the amount of time RNA polymerase is staying at one place, and that is somehow trying to nudge and keep on moving keep the RNA polymerase moving. And at the same time, this could also, if there is any misincorporation of any base, that is RNA polymerase has made a mistake, then TF2S is also involved in activating the RNase activity. RNA polymerase is about polymerizing, but it also has inherent RNase activity to remove, and this is especially the removal or RNase activity will happen when there is a misincorporation of a base. And all these things are orchestrated based on the phosphorylation status of this CTD domain of RNA polymerase. There is also another important point is FACT, facilitates chromatin transcription. That is what is referred to as FACT. So it consists of at least two different subunits, SPT16 and SSRP1. And it's supposedly what it does is, as the RNA polymerase is moving here, and it is involved in dislodging the nucleosomes here and also involved in formation of nucleosomes in the back. So how this apparently happens is that while the RNA polymerase is moving, the RNA polymerase in this case is moving in this direction. It is moving in this way. And this is the FACT, SSRP1 and SPT16, and those two proteins have a different function in the sense that their affinities are different. SSRP1 binds to H3H4 core histones, and SPT16 binds to H2A, H3, H2B histones. So when this particular nucleosome is dislodged from the chromatin, or in that place the DNA, these proteins, the FACT, SSRP1 and SPT16, are bound to H2A, H2B dimers and H3, H4 tetramers, and the RNA polymerase will pass by and they are immediately recruited in the formation of new nucleosomes behind the RNA polymerase. As the RNA polymerase passes, so we will have dislodging of histones on the front side and in the direction of the RNA polymerase, and nucleosomes forming in the back. So there is also other protein is thought to be involved that is called as spt6. So this is how the transcriptional elongation is thought to happen, and it is indeed a complex process. Thank you. See you later.